Hi, so I'm presenting my current master's project, and I'm also going to be talking about the gnatosome, so bear with me. But I'll be talking about is the dermal skeleton, which is a bit different. So first of all, these are the main groups of gnatosomes. I'll be mostly talking about acanthodians and scinctes. Acanthodians are already quite well researched, but I'll be using some new technology to further look at its dermal skeleton and scinctes. Scinctes is a bit mysterious in that we only have bits of fish scales for us to learn about, and people have suggested that it might be actually the earliest jawed vertebrate, and we'll see. So this particular specimen I'm looking at, for acanthodians, they're from the Silurian era, and for scinctes, they're from the Ordovician. So it's important to note that the scinctes predate acanthodians and all the other groups of gnatosomes, debatably. And the important thing about dermal skeletons is that for a lot of these early jawed vertebrates, they were covered in dermal skeleton, which later has been fossilized. And for some of the specimen, that is the only bit that has been fossilized. And if you're lucky, you get something like this, which is an acanthodian. It's actually really small. It's only like a couple centimeters in length. And then you can actually see that as a fish. But for something like scinctes, you only get like tiny bits of fish scales, and we don't really know much about them. So the more traditional approach to looking at dermal skeleton is using the scanning electron microscopy, which is a really good method. But one of the very big disadvantages is that you have to cut and grind at the fossil, so you're kind of destroying them in the process, which you really can't do with the more rare specimen like scinctes. And these are some of the images you can get using a scanning electron microscope. And you can see how detailed it is, and yeah, it's pretty cool. But what I mostly did was using Synchrotron Tomography. So it uses the power from a particle accelerator to generate high resolution X-ray scans. And you can use that you can construct it on the computer into like 3D, and like you can look at it from three different directions as opposed to one when you're just cutting it physically. And then you just put it in over here, and then do stuff with it. The magnifications we use is mostly 10 times 20 times, which gives you about a voxel size of 0.6 something. But then I also made some scanning 40 times, so it's a lot more detailed. So acanthodians, as we know them. So as you can see here, these are sketches made from data from scanning electron microscopy. And you can see what we call a positional growth in the dermal bone. And because this dermal skeleton is essentially bone, you can track its growth using the growth arrest lines from when the bone grows and kind of at different rates, depending on seasonal variation and like nutrition and stuff. And for acanthodians, what's really interesting is that they have what people call onion-like growth pattern, or if you watch Shrek, it's kind of like an ogre as layers, and then it it goes like that, and it grows from all directions. And you can also see that here. So this is one of the scans I'm using, and you can see the same pattern of a positional growth. One of the more interesting things we found, this is a scale from a skull plate of the acanthodian, which has not been described yet, to my knowledge. And then you can see a different pattern of growth. So it kind of is more directional. The younger thing kind of comes from here, and then you'll see it in the next slide. So it's a different pattern of growth. And And I segmented this data using a VISO. And you can see here that this is the inner bit from there, and then as the tubercle grows, kind of pushes it outwards. Yeah, you can see. Yeah, this is a view from the top, and this is a view from the side. So that is really interesting. And for Sciictus, so Sciictus, so there are only a few bits of specimens that we know of, and this is. And from this paper, like they highlight some of the main key features as like the growth lines here and the pulp cavity and what the authors think might be neural crest cells, which is debatable. And you can also see the amino layer on top. And you can also see that in our scans, and this is using synchrotron tomography, and you can also see the same growth lines and the pulp cavity, and it's a better preserved scale, so you can see more of the structure over here with the spongy layer and the amyloid on top. But we also found some of the specimens that have features that are a bit different. So this one and a lot of the other ones seem to have what resembles a neck canal, even though it still has the same similar patterns and others. And you can also see there's a bit of a separation here between the different layers. And for this one, it looks completely different, in my opinion. So the growth lines kind of encompasses the pulp cavity, and it kind of grows like that. So yeah, and I'm going a bit fast. So in conclusion, I can do skull plate pattern. So that's a bit of a novel thing, and it's kind of seems to resemble like a placoderm kind of pattern, which I had to read a bit more about. And for Sciictus, we found like there's two different types. One of them which hasn't really been described before, and then there are all these like growth patterns and neck canals that also I'll have to describe. So my project is about halfway done. So what I'm going to do next is especially for the Angathodian specimen, I got permission to grind them and observe them using SCM, which I'll then compare to the specimen I scanned in synchrotron. And also the interesting thing is we are trying to collaborate with the medical facilities at our university using this technology called wave propagation, which they use to follow the veins in your body. But they were going to use that technology to help segment the growth lines. So we are trying to make the process faster, because right now it's a quite labor-intensive process. Yeah. So I thanks to like all these people who provide us specimens, like Ivan Spamson, Daniel Goldberg, and also thanks to Phil Donaghy and Mike Richardson for supervising the project, also with Martin Rucklin. And yeah, any questions?
Any questions?
Do you know what the structure
So the question was what the structure of dentine in Sciictus is. So we think like we think the growth is kind of, there's a dentine layer and then everything kind of fills in between. So you can't really see from this picture, but yeah. And then we're not sure if that's dentine or semi-dentine or anything because it's from a scan, it's not from electron microscopy. But,
So are they kind of rounded or are they
they definitely look a bit more teardrop space. The question was what the cell space structure looks like. They look a bit more teardrop shape to me. But then I'm going to use the new data I made in February with the 40 times magnification to make sure, because yeah, from the 20 times it's not as clear.
Any other questions?
Yeah.
What do you think it is then?
What do you think what is? I think some of the structures resembles what you see in chondrichthyans and placoderms. So, and I'm hoping it does in the end resemble more of a more jawed fish because, yeah, better publication that way. But yeah, that's what I'm hoping. That's what I'm seeing. Sorry?
So we still don't know.
I'm like halfway through, so I'm not sure. Don't quote me on anything.
Any more questions? Okay, I'd just like to thank all our speakers once again.